Bienvenidos al seminario de hoy. ¿Me escuchan? No. Technical difficulties. ¿Me escuchan? Ahora sí, ¿no? ¿Nada? ¿Sí? Ya, ok, ahora sí, ya, mucho. Bueno, buenas tardes a nuestro seminario de la serie de Frontiers in Genomics. Bienvenidos a los que están acá, los que están en Zoom y en YouTube. Realmente es un honor presentarles a nuestro invitado del día de hoy, el doctor Satchidananda Panda, Sachi para los amigos. Sachi estudió una licenciatura en agricultura con especialidad en genética y cruzas de plantas en la India, en la Universidad de Orissa, y una maestría también en la India, en la Universidad de Tamil Nadu de Agricultura. Y después se movió a California en el 2001. ¿Ya? Ok. No. ¿Sí? Ok. Bueno, entonces, en el 2001 se mueve a California a hacer un doctorado en el instituto, en el Scripps Research Institute de La Joya. Y a partir de ahí ya no sale de California. Se mueve a hacer una estancia postdoctoral al Genomics Institute de Novartis en San Diego. Y regresa a La Joya, al Salk Institute, como profesor asistente en el 2004. Y desde ahí ha permanecido en el Salk como investigador. En el 2018 fue promovido como profesor en el Salk Institute. Desde muy temprano en su carrera ha recibido diferentes premios, reconocimientos a sus contribuciones. Entre ellas, la Fundación Dana le dio un reconocimiento por estudiar el sistema inmune con imágenes. Fue scholar de la Pew también. Whitehall Foundation también le dio un reconocimiento, ya en su carrera un poco más avanzada. Y en el 2021, Clarivate le dio un reconocimiento por ser uno de los autores más citados en el mundo. Como todos ustedes saben, el doctor Panda se ha especializado en entender cómo la luz, la nutrición y el ciclo circadiano controlan la salud y la enfermedad en diferentes condiciones patológicas. Y precisamente este estudio le ha valido grandes contribuciones a la ciencia. Y, por ejemplo, tiene más de 130 publicaciones en revistas de alto impacto, con más de 38,000 citas a las mismas. Está en el 99% de los autores más citados en el mundo. Y bueno, ha hecho grandes contribuciones en esta área. La contribución más relevante en los últimos años fue su publicación en el 2002, donde descubrió el sensor de la luz azul, los fotorreceptores que determinan esa función en el cerebro y que son los que hacen el setting del reloj biológico. Y bueno, pues hoy me da muchísimo gusto que el doctor esté aquí y nos va a hablar precisamente del ritmo circadiano, de la restricción de la dieta y la enfermedad y la salud. Thank you very much, Sachi, for being here.
Thank you. Thank you. Thank you for the kind and generous introduction. I'm really enjoying my time here, and thank you all the students who hosted me and kept me entertained with their scientific curiosity for the last couple of days. Why do we do science? Every day when I come to lab, I remind myself this thing that to live at our peak physical, intellectual, or emotional performance irrespective of age, gender, nationality, or health conditions, is a universal human right and also a human aspiration. And we as researchers, clinicians, whatever we do, our ultimate goal is to let everybody live at their peak performance at any stage of their life. In that context, I study circadian rhythm, and the ultimate goal is how to use the science of circadian rhythm to let everybody improve their health and increase their health span. So when you think of circadian rhythm, most people think of sleep-wake cycle, and those who are lucky enough to have gone to bed around 10 o'clock, your deepest sleep happened around 2 o'clock in the morning. Anticipating waking up, your body temperature begins to rise, your blood pressure begins to rise, and your respiration begins to rise. And then after waking up, within few minutes of exposure to light, your melatonin levels will likely drop, your cortisol levels are beginning to rise. And then your bowel movement is most likely in the first half of the day, and so is your peak insulin sensitivity. High alertness happens around middle of the day, and this is actually the time, late afternoon, when muscle performance is much better. So this is the time to go play soccer or run or do your physical activity. And then in the evening, melatonin begins to rise, your body temperature cools down, you can go back to sleep. So in fact, if you think of the best biomarker of healthy living, this is exactly how your body should react, should respond, and should show circadian rhythm. So in my opinion, having a strong circadian rhythm is both a manifestation of good health and is also the road to excellent health for a long, healthy lifespan. But if you think about it, circadian rhythm, which I'll get to very soon, that it's an intrinsic timekeeping mechanism, but it reciprocally interacts with three foundations of health, that is sleep, physical activity, and nutrition. And these three foundations of health also interact with each other in many different ways. And there's rich literature on how sleep affects metabolism, or metabolism affects physical activity, or even physical activity affects sleep, and vice versa. And all of these are also under the control of light and darkness. So then the question is, why should we study circadian rhythm? Why not all these individual factors that will actually give us that has been giving us many ways to improve healthy lifespan? The reason why I feel this is the time to study circadian rhythm is we have been living with the circadian rhythm since humans, Homo sapiens, evolved on this planet, maybe many hundreds of thousands of years ago. But over the last 200,000 years, after we mastered the controlled use of fire, we became a very different species. We are the only species on this planet that can control fire, and that's the distinguishing feature between early humans and other species. And now that led to modern industrial revolution, and we designed our anthropogenic world without paying attention to these rhythms. So that's how we came up with shift work and other stuff, because staying up late into the night is the biggest survival advantage and competitive advantage you can have against your enemy. Whether it's winning the war, whether it's exploring to a new era, new place, discovering new islands, new continents, or navigating through the night, all of these require you to master the evening. But at the same time, we also figured out that this chronic circadian rhythm disruption can increase risk for many diseases. So then the question is, how can we redesign our anthropogenic world with the knowledge of circadian rhythm so that we can continue to sustain the wealth and all the comfort that we have in modern industrialized era, and at the same time we can be healthy. So the circadian rhythms, as you might think, there is a circadian clock in the brain that reset, rejuvenate, and repair our brain as we go to sleep every single day. So similarly, the rest of our body also has cell autonomous or tissue autonomous circadian clock, and the brain's function is to act as a conductor of this rhythm, so that as a whole the body produces daily rhythms in physiology, metabolism, behavior, as well as even rhythms in microbiome and their metabolites. And this circadian system is connected to the outside world primarily through light received through the eyes. And the reason why the circadian clock has to be in trend to light-dark cycle is in nature. As seasons change and day length change, the organism has to anticipate change in day length and wake up or go to bed at the appropriate time. And at cell autonomous level, as we know now, circadian clocks are composed of transcription translation feedback loops, in which clock and BMAL, these two transcription factors, play a central role by binding to the cognate site E boxes CACGTGA to drive transcription of cryptochromes and period genes and also many other genes, which in turn inhibit the function of clock BMAL, and that produces rhythms in cryptochrome and period. Clock BMAL also bind to the promoters of ROR and reverb class of nuclear hormone receptor. Those are key regulators of metabolism and immune system. And ROR and reverb act as activators and repressors of BMAL1 respectively and produce rhythms in BMAL1. So as you can see, a bunch of transcription factors are essentially interacting to produce their own rhythms. So it's not surprising that this circadian system also drives rhythmic transcription of many other genes in a tissue-specific manner. So then the question is, if we know some of the molecular aspects of circadian rhythm, so the reason why we do research is to figure out what are the key biological principles behind a phenomena and then leverage those biological principles to develop new lifestyle or new drugs or new therapies. So here the question is, what can we learn from, what are the biological principles here? As I mentioned, the clock has to be entrained to the outside world through light and darkness. And many years ago, we knew that almost for last 75 years, starting from 1923, people knew that although rods and cones in our eyes are essential for seeing the outside world or visual system, they are dispensable, completely dispensable for entraining the clock. So that means there must be a missing photoreceptor that we didn't know. And when I was a postdoc, that's the time when 3 different labs independently discovered the role of melanopsin. The opsin from melanosomes, frog melanosomes, that's why named melanopsin. It's expressed in very few, hardly 5,000 retinal ganglion cells in human retina, or 2,000 retinal ganglion cells in mouse retina that sense light and directly, and these neurons are monosynaptically connected to the master circadian clock in the suprachiasmatic nucleus in the hypothalamus. So mice that lack melanopsin cannot appropriately reset the master circadian clock. So by learning that biological principle that melanopsin is a dominant photoentraining signal and it's most sensitive in blue light zone, that now can be used to understand some aspects of circadian disruption. So for example, our ancestors who have no access to electricity or evening bright light, melanopsin is also less sensitive to light. So that means candlelight, firelight cannot fully activate melanopsin. And in that way, our ancestors their melanopsin system was not activated in the evening. The melatonin levels began to rise at the right time and they could go back to sleep between 9:30 and 10:30 p.m. if sunset happened around 6 p.m. And during daytime, melanopsin also requires bright light to be activated, and melanopsin also raises alertness and reduces depression. So as a result, as our ancestors got out of their hut or their small houses and spent most of their time outdoor looking for food, then this bright light exposure activated melanopsin, synchronized the brain circadian clock, reduced depression, improved alertness. So now the question is, what is happening in the modern world? In modern world, we actually spend a lot of time indoor, and in the evening we're exposed to bright screen and bright light at night that activates melanopsin, which reduces melatonin, and we have poor sleep. And during daytime, we are stuck in dimly lit room like this. For example, inside this room, you are getting somewhere to between 200 to 400 lux of light maximum, whereas if you just walk outside, even without standing under direct sun, you'll get 5,000 to 10,000 lux of light. And that's the amount of light, at least 1,000 lux or more is optimum to activate melanopsin system. So as a result, we go from insomnia or poor sleep at night, and daytime we try to stay awake by consuming coffee in every 2 to 3 hours. And if it continues for a long period of time, then that increases the risk for many affective disorders, from irritation, ASD, ADHD to even PTSD, mania, and bipolar disorder. So as a result, now this new biological principle has started what is called lighting revolution, or can there be healthy lighting? And this is a really turning point in the history of human, because until the discovery of melanopsin, lighting was considered as a comfort and safety. We lit up places to feel safe or to just be active. For the first time, this discovery is now changing how we should light up our classrooms, our space stations, old age homes, hospitals, and classrooms, so that we get more melanopsin activation only during daytime, but we reduce that melanopsin activation by reducing blue light exposure at night. And in that way, this is changing almost $30 billion of lighting industry to come up with human-centric lighting. So then the question is, for lay people and everybody like us, how much light is healthy? So for example, lighting to treat depression, seasonal affective disorders, or ADHD is around 10,000 lux of light for approximately an hour. So that means being outdoor, not under the sun, because under the direct sun you'll get 150,000 to 250,000 lux of light. And an average person should get 1,000 lux of light for an hour. So that is equivalent to having your lunch or breakfast next to a large window, or ideally outdoor. And in the evening, you should have less than 20 lux of light, at least for 2 hours before going to bed. If you want to work, then you can use table lamps. People always ask me, hey, my phone turns yellow or orange, is that going to improve my sleep? The answer is no. It's just telling you that you should wind down and go to sleep. So for example, just like an alarm clock wakes you up but doesn't make you energized to get out of the bed and start running. So similarly, this light change that happens in your smartphone is a nudge, is a reminder. It's like an alarm clock to wind down and go to sleep. Here is also another extreme case of using that biological principle. So this study was actually done in Mexico City, and we know that 1 in 10 live births in Western countries, including Mexico, is a premature baby. And preemies are essentially put into NICU, neonatal ICU, where they're exposed to continuous light. And the idea is continuous light can disrupt your circadian rhythm, and babies are actually having a good circadian rhythm. Although they cry and sleep in every 3 hours, they do have strong circadian rhythm. So the experiment was very simple: to keep the babies in continuous light as standard of care, or to simulate light dark cycle by having 250 lux of light during daytime and covering their head with a kind of a cloth helmet, so that the eye gets only 27 lux of light in the evening. And by doing this, what they found was these premature babies on LD simulation can grow much faster and can get out of NICU on an average 13 days before the standard of care. So just imagine 1 day of adult ICU or NICU costs around $10,000 to $15,000 in the US. So 13 days of NICU care in 385,000 premature babies in the US will pay for all the circadian research ever done, and will be done in the next 50 years. So these are some of the huge impacts of learning biological principles of rhythms and then applying it. The other example is we know that during puberty, for some reason, people, both males and females, they become more sensitive to light, or maybe that's the time when they get more used to caffeine and they stay awake late into the night, and in the morning they're not awake enough to go to school around 7:30. So in collaboration with Horacio de Iglesia, he did a very nice, impactful study where he delayed the high school start time in 2 different high schools in Seattle, and found that by delaying high school start time by 1 hour, the students got 34 minutes of extra sleep, 4.5% increase in median grades, and their tardiness went down. And that particular study, along with many other studies, gave credence to this law that was passed in California, that no middle school or high school in California can start before 8:30 a.m. in the morning. This is another example. By learning biological principles of circadian rhythm, we can actually change public policy and improve the health of at least 5 million students in a state. and hopefully this will have ripple effect. So now I'll switch gear and I'll talk about how diet and clock interact. This is a seminal paper from Joe Bass' lab, which essentially showed that high-fat diet-induced obesity, that's a model used in 11,000 papers in PubMed, actually disrupts circadian rhythm, because these mice, instead of eating 18%, 15 to 18% food during daytime, they actually increase their food intake slightly during daytime. They eat up to 30% of their food. For most people, this doesn't matter, because the non-circadian researchers would say, only 15% increase in food, so that's few, less than a gram increase in food intake. But then that begged the question for us, how much of this diet-induced obesity and related disorders are due to diet, and how much of it is due to mistimed eating? So we did a very simple experiment in which we had 4 different groups of mice, normal chow ad libitum, normal chow time restricted, high fat ad libitum, and high fat time restricted. They were in this regimen for 18 weeks. So the energy consumption by all these 4 groups were equivalent, and the activity was also equivalent. The only difference was the time restricted group was more active towards the end of the feeding period. The ad lib group was more active at the beginning of the feeding period or evening, and the respiratory exchange ratio or the metabolic flexibility improved under time restricted eating. And to our surprise, that high fat diet induced obesity that's found in high fat, this FA group, was almost prevented. This is very important, prevention, not cure, in high fat time restricted feeding group, because the body weight was not significantly different from normal chow ad lib fed mice. And this study was repeated at least 3 times before we published it. So that's why we have 4 co-first author in this manuscript. At that time, it was too good to be true. I used to give presentation and people used to say, you must have done something wrong. Maybe the mice didn't eat. And then if you want to be very smart in metabolic research, you want to show off, then you ask, have you equalized the 3rd law of thermodynamics? The bottom line is there is not even a single mouse paper in the entire world that actually showed the 3rd law of thermodynamics. So when somebody asks you that question, that means that person doesn't trust your result and wants to show off by invoking some law of physics that has never been equated in metabolism research in mouse or human even. So this was a quite stressful time that time, because for a couple of years nobody believed us. But then Amandine Che also came, and then we said, okay, let's try this in many different diets. So he had 500 plus mice, where we asked whether if the mice were already obese and if we put them on time restricted feeding, will they lose weight? And this is very important, because there are a lot of preventative measures, preventative aspects of disease, but they're not usually therapeutic. Best example, smoking cessation reduces the risk of lung cancer. But a lung cancer patient who continues to smoke, smoking cessation will not cure cancer. So you got to also ask whether the intervention that we are suggesting can reverse the disease. And the bottom line was, yes, it could reverse, but it doesn't have any legacy effect, because mice that are fed time restricted feeding for 23, 26 weeks, if you release them to ad libitum feeding, they quickly gain weight. So there is no legacy effect. Mice that ate normal chow, you don't see much body weight differences, but they actually maintain their muscle mass or increase their muscle mass under time restricted feeding. And time restricted feeding effects are modulated by sex. So for example, female mice don't gain too much weight, so that's why they don't also, time restricted feeding doesn't have much effect on body weight in female mice, but in male mice, since high fat diet causes obesity, there is a prevention of obesity. But overall, all the other effects are more or less reproducible between male and female. The magnitude is different. Then in the last several years, we have seen many benefits of time restricted feeding in multiple organs, in mice, flies, rats, even in some cases humans. And these benefits likely arise from multiple organ systems. For example, starting from liver and plasma triglyceride, which is largely from liver, to acid reflux, which might be from digestive system, and female reproductive lifespan in at least one study. Motor coordination might come from neurons. So that's why we thought, what is the global impact of time-restricted feeding on different organ systems? So we collected 22 different peripheral organs and brain regions from mice that are exposed to ad libitum or time-restricted feeding for only short period of time, 6 to 7 weeks, in which they don't gain too much body weight. So the time-restricted feeding also doesn't have too much impact on body weight. And then we did 1,000 plus RNA-seq because we also collected time series data to test another hypothesis, that is, does time-restricted feeding improve the amplitude of circadian clock in multiple different organs? So to address that question, you have to do time series data analysis. I'll not go into detail, but if we just ask what most people tend to ask, what are the genes that are differentially expressed between two different conditions? The bottom line was the organs that showed the most differences were the adipose tissue, brown adipose tissue, white adipose tissue, two different types. And then the next one was the digestive system, but surprisingly only duodenum and jejunum, but not too much differences in the ileum. And then of course the next ones are the HPA axis, hypothalamus adrenal, and then liver, heart, kidney, muscle, and stomach. So those are the differentially expressed genes if we take all those 12 time points as replicates and then just do a very simple differential expression analysis. Then you can ask many different questions. For example, you can ask, what are the common genes that are upregulated or downregulated in multiple tissues, and what do they do? The bottom line is time-restricted feeding upregulated stuff or pathways or genes that are involved in RNA processing, fatty acid catabolism, protein processing in the ER, ribosome and lysosome. So that means turnover of RNA and protein is improved under time-restricted feeding. And in terms of downregulated genes, it's essentially oxidative stress, excessive immune system activation, and branch and amino acid degradation. So those some of the pathways that are common. We can also ask what happened to the cycling genes. If we stack off all the cycling genes in multiple different tissues, what we find is under time-restricted feeding, there is robust coordination of rhythms in multiple organs. As a result, there is a huge peak of transcriptional cycling, and all these transcripts reach their peak expression level late in the afternoon before feeding begins. So that's the anticipatory aspect of circadian rhythm. And there is another peak of activity towards the end of the feeding cycle. And if we ask what are the pathways that are upregulated during fasting phase or feeding phase, then some are expected. For example, autophagy, DNA damage repair, but surprisingly cell cycle regulation also popped up. And again, RNA and protein quality control essentially goes up only in the fed state. So now with this data, you can also come up with new hypotheses. For example, if we look at genes that are involved in lipid metabolism, starting from lipid absorption, then its metabolism in the white adipose tissue, reuptake in the liver, and then production of VLDL particles and cholesterol breakdown, then we saw quite a few genes, critical genes, that become more cyclic or they become more coordinated. For example, in EUR, they are more coordinated under time-restricted condition. So that led to the hypothesis that maybe time-restricted feeding also improves dyslipidemia because at the end of the day, what we know now is globally the #1 cause for death or disability is heart disease, and heart disease is mostly driven by two different factors. One is high glucose and one is dyslipidemia or excessive triglyceride or LDL particle or VLDL particle. And most of the current existing drugs to treat dyslipidemia, whether it's statins or even PCSK9 inhibitors, those depend on the functional state of LDL receptor. So those individuals, there is at least 1 in 160 people who have a mutation in LDL receptor. They don't respond to any of these therapies. So that's why it's very important that if you want to show a beneficial effect of a treatment to address dyslipidemia, if it can work on LDL receptor knockout mice, that's even better because you can actually address the ones who are resistant to statin therapy. So we put LDL receptor knockout mice on high fat, high cholesterol ad libitum or time restricted, And as seen in B6 mice, these mice eat the almost equivalent amount of food, but they are protected from weight gain. And they do so reduction in serum triglyceride, serum cholesterol, and also if we do fractionation of the lipid profile, we do see reduction in VLDL and chylomicron remnant, IDL, LDL, but there is no improvement in HDL. This is important to know because we always think that many interventions can reduce LDL and increase HDL, but time restricted feeding does not change HDL levels. And at the end of, I think it was 12 weeks experiment, we also found there is a 32% reduction in atherosclerotic plaque in aorta, and by different measures we found similar results. However, this effect is not found in ApoE knockout mice. ApoE is required for reabsorption of LDL particle from blood, getting into the liver and metabolizing that. And since ApoE knockout mice cannot reabsorb all the LDL particle from plasma, we think that this liver metabolism or VLDL and LDL or cholesterol is important for the improvement in dyslipidemia in time-restricted fed mice. So just like we expected, the ApoE knockout mice do not show improvement in atherosclerosis or LDL or IDL levels. And I'll not go into the detail, but we can actually ask whether there is a defect in absorption of triglyceride from the intestine, its lipoprotein lipase activity in circulation or in the adipose tissue, its reabsorption by liver metabolism, and then VLDL secretion. So the take-home message is the VLDL secretion is the one that is reduced under time-restricted feeding in both LDL receptor knockout and ApoE knockout. But since ApoE knockout mice don't take up too much LDL into the liver, it doesn't actually matter too much. So the bottom line is the mechanism seems to be the liver metabolism of LDL and cholesterol. So we did a lot of gene expression profiling and metabolomics, and that essentially reconfirmed what I just said. And at the same time, we also looked at the fecal loss of cholesterol, because what we found is in the liver, cholesterol is broken down to bile acids a lot more under time-restricted feeding. And since bile acids and cholesterol both can get into the intestine, then the question is, are these mice losing a lot of cholesterol and bile acid? And the answer is yes. These mice under time-restricted feeding actually lose a lot of cholesterol in their poop. So similarly, just like the cardiovascular disease or the dyslipidemia, another cause of death or disability is the problem with kidney, because hypertension is partly contributed by kidney. And also in advanced stages of diabetes, people actually lose kidney function. There is chronic kidney disease. Nearly 37 million people in the US adults have CKD. The same numbers actually have type 2 diabetes, and the main cause is diabetes, high blood pressure, cardiovascular disease. And the diagnosis is albumin creatine ratio and then glomerular filtration rate. And in extreme cases, people do biopsy for fibrosis to see whether kidneys have fibrosis. And there are essentially different stages of kidney disease, but in mice, you can actually do two different models. One is unilateral ureter obstruction. You tie up one kidney so that the kidney is damaged very quickly within few days. And then the other one is inject excessive amount of folic acid into the mouse, and that folic acid is toxic to kidney, and kidney failure will happen. So the question was very simple. Can time-restricted feeding improve the health of kidney in these two different models? The nice thing about unilateral ureter UUO is one kidney is healthy, the other kidney is diseased. So within the same mouse, you have a control. And again, the experimental design is same. We can have a preventative model or a therapeutic model. And just like in previous studies, we also see these mice consume the same number of calories, but they reduce body weight, and most of the body weight reduction is due to reduction in fat mass and not actually changes in lean mass. The proportional lean mass actually goes up. So the bottom line, does it reduce blood urea nitrogen, which is a marker both in mice and humans of kidney disease, and creatine? And then the answer is yes in both, at least in the folic acid model, where both kidneys are affected, time restricted feeding improves or reduces this blood urea nitrogen. In the UUO model, one kidney is actually enough to function so that your bone level, blood urea nitrogen levels don't change. And that's also the basis why people who need kidney transplant, they just need one kidney, not two, and the donors can still survive well with one kidney. But still, in the creatine levels, we do see improvement in creatine level. Then the question is, okay, so what are the benefits? Can we see it as in a histological state and in gene expression state? So we do different changes. For example, you can do simple H&E staining to see whether there is damage. Renal damage, there is a marker KIM1 that will also give you more specific renal damage. There are inflammation marker, for example, F480 macrophage infiltration, and then fibrosis markers. And you can also check for mitochondria because many of the kidney diseases affect mitochondria function. So in the next few slides, you don't have to, I won't have time to go through the detail, but then the bottom line is in most cases, time-restricted feeding improved kidney function as evidenced by H&E staining, simple H&E staining. This is the vehicle, that's FAN, and then this is the time-restricted feeding group over here. They look much better, more healthier than the folic acid or UUO model. Then same thing with the KIM1 staining, and then finally we also see reduction in kidney fibrosis in kidney injury under this model. The inflammation also goes down, and if we look at the inflammatory signature, for example, if we look for F480 staining, that also significantly goes down in UUO model and also in the FAN model. And mitochondria function improves. We also did gene expression profiling and essentially found that time-restricted feeding improved the CKD signature or chronic kidney disease signature. So with that, I think now there are enough publications to support that in addition to quality and quantity of nutrition, the timing of nutrition also matters. It doesn't mean, because many people, they ask me, so do you think that the quality and quantity doesn't matter? I said, no, they do matter. But this is another, if it is a three-legged stool, then this is another leg. Okay, so also this is now nicely replicated in calorie restriction studies that mice, in many calorie restriction studies, they're actually inadvertently put into time restriction, because in mouse and rat calorie restriction studies, mice and rats are given 1 meal a day and they finish that food within 2 to 8 hours, depending on the vivarium. And in fact, this beautiful paper, very systematic paper from Joe Takahashi lab, showed that if you combine time restriction with calorie restriction, you can increase lifespan by 35%. Whereas if you have only calorie restriction, then you increase median lifespan by 10%. So that's very profound. That essentially says that both timing and calorie restriction do matter. And in fact, all the CR studies are also done on normal chow or healthy diet. So the bottom line is yes, those three do matter. So there are a lot of now attention to time restricted feeding, as seen in many reviews and also in popular media. And then the question is, how do we go from mice to humans? So a few years ago, when people are questioning whether time restricted feeding is relevant to human health because people eat, we always think that we eat 3 meals a day. Thank you, Gustavo. Have a safe trip. People eat 3 meals a day within 12 hours. We asked, do they really? Because unfortunately, in the last 150 years of nutrition research, not even a single paper, just imagine 150 years of nutrition research, not even a single paper accurately described the day-to-day variation in nutrition timing. The gold standard in nutrition research is 24 hours dietary recall. And it's essentially asking people, what did you eat yesterday? And But from circadian point of view, we wanted to know how much day-to-day variation we have and how many meals people eat. So we asked people to take a picture of whatever they're eating or drinking, and then second click they will save the picture, and we would assess whether there was a calorie containing food in the picture. And we collected this information for 3 weeks because we thought in 3 weeks we'll have three sets of weekdays, 3 sets of weekends, and then that will give us very good idea of how day-to-day variation in meal timing occurs. And then we can figure out what is the window of time when a person is likely to eat, because one day of information will not give you that data. So this is 3 weeks of data. The blue is weekday, the red is weekend. As you can see, there is change in eating time, but if we combine it and put it around the clock, then this is how 3 weeks of nutrition containing food looks like, where top is 6 a.m., bottom is 6 p.m., and then this is 156 people's data. None of them was a shift worker, or no one was a student. So we excluded students and shift workers because students are almost shift workers. So the bottom line is, yes, as expected, the number of events or number of logs, people logging, peaked around noon and evening time when people eat lunch and dinner. But the amount of calories guesstimated from each picture actually peaks in the middle of the night when people get up and eat something, and they're eating a big bowl of cereal or ice cream or something else. And if we ask what is the time interval within which a person is 95% likely to eat, this is very different from asking when did you eat your breakfast yesterday, when did you eat your dinner yesterday. But this is the interval when which a person is likely to eat within 2 to 3 weeks interval. Then what we found is nearly 50% of adults eat for 14 hours 45 minutes or longer. Less than 10% of adults actually eat within 12 hours or less, and that probability band is 12 hours or less. So with that, we have quite a few studies going on to see whether we can modify people's behavior. But in broader scale, if you think about what affects our health, it's the health state. What is your current health? Because sometimes your health state might prevent you from adopting healthy lifestyle, from many other things. And then health awareness, are you aware of what is actually healthy lifestyle? If you don't know what is healthy lifestyle, then you are less likely to follow it. Environmental exposures, genetics and epigenetics, and your current lifestyle. These are the major factors that affect. And in many of these cases, many of these are non-modifiable, but lifestyle is something modifiable only if the mindset is there, awareness is there, and there is willingness and opportunity to change health. So when you think of the culture of health, has 2 components: letting people know about what is healthy and giving them the opportunity to practice what is healthy. If you give them the knowledge alone, for example, if you tell people that reducing calories is good for you and improving your nutrition quality is good for you, but they don't have the opportunity to read the nutrition label and do the math every single time, or they have no access to healthy food, then you lose the culture of health. So what we do is we collect through this app. Now this app has been expanded, so we can combine it with clinical research so that we can get health state information from clinic visit, and will also increase health awareness and mindset through surveys, blogs, videos, and nudges. And we can modify their lifestyle, or people can modify their lifestyle. From geolocation or work plus, we can get some information about environment and exposure, but we are not changing anything with genetics and genomics. So the app actually does let people self-monitor their 3 foundations: exercise, sleep, and nutrition. And with that, we also combine that with sometimes activity watch or continuous glucose monitor. So you get a very rich data set of what people are eating, when they're eating, and how that is reflected in their continuous glucose measurement. And that leads to different kinds of report. For example, you can just use a simple word cloud that tells what this person actually logged in the last 2 to 3 weeks. And then feedogram, you can also combine that with continuous glucose monitor and see how the person is responding. And you can combine, we do combine and give this very comprehensive clock plot to people. So this is going on in at least 12 different studies, and many of them are published, many are ongoing. But at the same time, we have a public-facing app where anyone, anywhere in the world can download the app and can self-monitor what they're doing. And through that, we have collected nearly 9+ million food record. Actually, right now it's 9 million. This was an older slide. And from there, we can use machine learning algorithm because they're using their own natural language to describe what they eat. Sometimes there are typos, sometimes there are natural language differences. For example, people in the US will say coffee, some people will say cafe, and there is some typo that will say COFFFEEE, so there are many other things. So by NLP, we can extract and then we can get almost 90+% of those meals or records accurate. So then the question is, what can we learn from there? So for example, there is a participant that is reporting a meal log, and in that meal log there are many components. On the left side, somebody can just say meal or last meal or first meal, so of course we cannot get any meal information. But on the right side, somebody who is actually describing the meal pretty well, we can get a lot of information. One simple question is, how many, we always do many research and we say, if you eat a lot of different types of food, then you are healthy. If you have a high food diversity, your gut microbiome is also rich. Simple question, how many different type of food or different food a person eats in a week? Nobody has figured that out, right? There's no publication, not even a single, that says how many different types of food a person eats in a week. So we asked that question. So we actually asked for 2 weeks because, what we ask is, suppose say I reported a lot of data and I might have eaten rice 20 times, but that counts as one because that's one type of food. I might have eaten tortilla 1 time, that also counts as 1. So if we add up how many different types of food a person eats, then what we find is the top quartile can eat up to 75 or 80 different types of food and beverages within 2 weeks, whereas the bottom quartile eats 20-25 different food, unique food within 2 weeks. But if we guesstimate how much of calories comes from, how many foods account for 75% of calories intake, it's actually a very small number. It's only 12 or 13. So if you want to change somebody's food intake behavior, majority macronutrient change, then you got to change only 2 or 3 food that the person most frequently eats. So these are the kind of stuff you can do from the app. If we extend it, because there are a lot of people who actually continue self-logging the food for up to a year, and if we take it up to 85 days or 90 days, then what we find is the top quartile eats almost 250 different types of food, and the bottom quartile eats 55 different types of food within 98 days. So you can use the also app because people are logging everything. Sometimes they do also log medications. So we have at least 560 different medications and supplements in the library. And whatever they log, we can, because sometimes the log is a commercial name or a trade name, we can translate it back to what is the active ingredient, and we can ask. Very simple thing. What time of the day people take medications for hypothyroidism? People who take thyroid medication, it's very common that they wake up, take the medicine, and then wait for at least 20-30 minutes before their breakfast. So it's not surprising that that peaks around 6 a.m. in the morning, or between 6 and 6:30 a.m. in the morning. But if we think about the frequency of medication to treat your constipation, it's, of course, people get up, try, and then they try the constipation medication. So it's 8 o'clock or late at night if they are constipated throughout the day. And diabetes medication is mostly in the morning. And then acid reflux, people get up, they have acid reflux, heartburn, and that's also they take very quickly within a few minutes of getting up. Cholesterol medication, hypertension medication is very surprising because we know that many hypertensive medication, if taken at bedtime, reduces the 5 years risk for heart disease or stroke. But surprisingly, nearly 3/4 of people take the hypertensive medication in the morning. So this is a very nice example of how you can nudge people's behavior, telling them what the recent scientific finding is, and hopefully they can change the behavior of medication timing and improve their health. So you can get all kinds of information, for example, contraceptives, and then insomnia. You hope that people are taking melatonin in the evening, not in the morning. Of course, the ones that are taking in the morning, they're shift workers. So they're coming back from night shift. So that's why they're taking that sleep medication in the morning. So the bottom line is this app is actually telling us a lot of stuff. We have nearly 29 publications from this app, and then we are expanding. We just released a more robust version of the app so that we can monitor at a time 50,000 people can simultaneously get on the app. So what do we do? What is the take-home message? If you have to take some take-home message, then the take-home message is try to be in bed for 8 hours, because all sleep researchers agree that all adults should try to sleep at least 6.5 to 7.5 hours. That large epidemiological studies involving 5 to 10 million people have shown that sleeping between 6.5 to 7.5 hours is associated with least mortality and morbidity. And then after waking up, wait for an hour after waking up to have your first meal, because that's when your cortisol levels peak, your melatonin levels is still high. And then have a consistent breakfast, because changing your breakfast time also changes how your circadian rhythm acts, works, resets. And then after breakfast, try to eat everything within 8, 10, or maximum 12 hours. Because if you go beyond 12 hours, then you get to eat very close to your bedtime, which may not be good for getting a good night's sleep, and also bad for glucose regulation. So that's the bottom line of what we are doing. And we have a new line of research, but if you want to hear a little bit, then I will give you a gist. One thing, everybody thinks that calorie restriction is good, exercise is good. Can you combine calorie restriction with exercise? And if you combine, what happens? In fact, nearly 40% of athletes reduce their calorie intake for either sports purpose, because they want to qualify to a lower weight group, or inadvertently they reduce their calorie intake. And that can lead to what is called relative energy deficiency in sports. And this is a syndrome. This is newly recognized. Nearly 40% of our athletes do that. And many people in the general population also intentionally reduce caloric intake and increase their exercise. And that can lead to multiple problems with their health, and I'll get to that. So how do we do that in a mouse model? There is no mouse model of exercise with calorie restriction. What we do is we put mice on running wheel, and then they have to run 10 meters or so before they get a food reward. And progressively you can reduce that reward so that they will continue to exercise the same number of rotations, but they are progressively getting less and less rewards in every week. So in that way, you can create energy deficiency without changing exercise level. So here the female mice exercise the same amount, but they eat less and they also lose weight. The male mice exercise to the same amount, they reduce food intake, and they actually lose more weight. And then if we ask what are they losing, they lose all three. They lose lean mass, they lose fat mass, they also lose the rest of the other things. And that happens in both male and female mice. Then if we ask what organs are affected, then what is interesting is in female mice, mostly the uterus and ovaries significantly reduce their weight. Kidney is also affected. In male mice, it's kidney and testis. To some extent, heart and spleen are also affected. Then they all lose muscle mass. And then if we ask what happens to the bones, because many of these athletes actually have fragile bone, they get mostly bone injuries, they have micro fractures, and those accumulate with time. And what we find is, yes, these mice also reduce their bone density, and particularly the cortical bone density, which is very important. That's where many of the fractures actually happen in athletes. And then if we ask what happens to a sleep-wake cycle, because many athletes actually report that their sleep is fragmented. And in fact, that's exactly what we find, that in energy deficiency, the energy sufficient mouse is mostly night active, whereas the energy deficient mouse is mostly day active, because the I switched that. The energy sufficient mouse is mostly sleeps in the daytime, active in the nighttime, and then that changes in the energy deficient mouse. They actually have fragmented sleep. They sleep and wake up in every few hours. And that's also reported in many athletes who go through relative energy deficiency. We also asked what happens to the estrous cycle in female mice, and we do see the female mice get mostly stuck in the diestrus. And we know that in humans also, many athletes either become amenorrheic or they have irregular menstrual cycle. Then the blood glucose levels drop, as you would expect, and then the corticosterone levels skyrocket. So in summary, we have now a nice model of relative energy deficit syndrome in mice, and hopefully we'll learn whether this has any epigenetic mark and whether we can reverse it, how much we can reverse it, because in the context of Olympics this year, and we know that nearly 30% Olympic athletes actually suffer from this. So with that, I'll stop and take any questions. Thank you.
Hi. Okay. Thank you for the seminar. I was wondering, what about the circadian cycle of the microbiome? I mean, how does it relate and interact with the host circadian cycle, and how they affect each other?
So unlike the cell autonomous nature of circadian clock, I don't think any microbe has a cell autonomous circadian rhythm that is self-sustained. So most of the microbial rhythms that have been described so far is, I would say, driven rhythms, because with metabolism there is change in microenvironment in the gut and the digestive system. So with that drives the changes in gene expression, as in metagenomic expression of many microbes that are almost symbiotically associated with the ileum, jejunum, and also there is circadian rhythm in microbial population itself. That composition changes throughout day and night. So that's how, and then there is rich literature on how microbial changes can also affect host population, host circadian rhythm. That's it. You can see a lot of references on that topic. Thank you.
Thank you for the talk. I was wondering about the data that you showed about the kidney. These mice, were they fed a high-fat diet that I didn't get?
They were fed a moderately high-fat diet. This is 43% fat and sucrose, because that sucrose and fat combination causes more kidney damage than regular. Yeah.
So what happens to mice that are time-restricted fed, but they are on a controlled diet?
Yeah, I think the benefit will be even better. But the folic acid injection can work much better with the high-fat, high-sucrose diet to cause more inflammation. So
yeah. And then, do you think that we can leverage all the knowledge that we know about the benefits of time-restricted feeding to improve the lifestyle of people that are night shift workers or that work at night?
Yeah, we did have actually a randomized controlled trial on firefighters in the US, in San Diego, who do 24-hour shift. So they do on an average 3 to 5 calls every night. So that means they're waking up 3 to 5 times. Maybe actually more than that, because they wake up, but if the engine is not called, then they don't go, but they wake up anyways. And in those individuals, we have found that time-restricted feeding improves their blood sugar, blood cholesterol, and dyslipidemia among firefighters who are unhealthy to begin with. Those who are healthy, they stay healthy. We haven't actually looked at strictly night shift workers because of many logistics issues too, because the night shift workers are less likely to come to clinic for their regular checkup and other stuff. But yeah, our group will actually start a night shift nurses study soon. End of the year, we'll start.
Thanks.
Very interesting.
Thank you.
Congratulations. You mentioned something about brain functions, not in the hypothalamic function, but you also mentioned functions like that disturb, like Alzheimer's disease, cognitive functions. And we know that the hippocampus also have rhythms and this stuff. Do you know what is the influence by time restricted? Because we know that the suprachiasmatic nucleus is mostly influenced by the light dark cycle instant to nutrients. But what about these other brains areas, limbic areas, and all this stuff? Do you have an idea of this?
Yeah, there are actually very nice publications from Pierre Chamoun's lab showing that timing of food actually affects all the rhythms in the brain except the SCN. Even PVN, DMH, which are only few millimeters away from SCN in the mouse, those track timing of food, not the light dark cycle. So along that line, there are also studies done in Huntington disease mouse model and Alzheimer's disease mouse model showing that time-restricted feeding improves sleep and reduces the severity of the disease. And in fact, only last few months, the Alzheimer's disease time-restricted feeding paper was published in Cell Metabolism from Paula Desplat's lab in UCSD.
Okay, but you know, if there is an experiment, for example, for memory formation or you know, deep cognitive function like decision taking or something like that. There is nothing with a paradigm in feeding restriction. Do you have an idea? Or even in people, do you know if there is a relation or with because you mentioned Alzheimer, ADHD, if there is something that improves in this deep cognitive functions with time restricted. I don't know if maybe this is new something, but I was wondering if there is an experiment just dedicated for that, you know, to see, for example, in mice or on rats, if restriction feeding improves this.
So actually,
something new maybe.
In humans, there are studies showing that managing light in the evening is beneficial for reducing the severity of ADSD and ASD.
Oh, okay.
So by managing light, you're also promoting sleep or improving sleep, and that improves. There are also studies showing, longitudinal studies from UK showing that babies up to the age of 3 or 5 years, if they are put to sleep at the regular time, then they have a less risk of developing obesity and also some of the affective disorders later in life. Then in terms of time-restricted feeding and ADSD or ASD, there is not much studies there because of obvious difficulties, but then there are at least 2 studies starting showing time-restricted, testing the effect of time-restricted feeding on reducing the severity of bipolar disorders.
Okay.
People with bipolar disorders who are treatment resistant. Yeah. So at least there are 2 studies that are beginning. That's based on studies showing that timing of food intake actually affects this affective state in humans in controlled environment. That was, I think, from Frank Shear's lab. And based on that, there are 2 time-restricted feeding studies on bipolar disorders.
So finally, maybe all the clock control functions should be influenced by time restricted, maybe, probably, I don't know. But,
well, not directly, sometimes indirectly, because, for example, what we see typically, in our studies, what we do is, we don't give them a strict, like, you got to eat between 8 a.m. and 4 p.m. What we tell them is, based on your own lifestyle, pick a 10-hour interval not 8 or 6, 10-hour interval that you can stick to, but your last meal should be 3 hours before bedtime. So in that case, what we see is people's self-reported sleep quality improves. So I don't know, and I cannot say whether time-restricted feeding alone improves affective state or brain state, or it's the improvement in sleep quality that improves. So it's too early. Means if I'm selfish, I would say yes, time-restricted feeding will improve brain health. But actually, what we do see in both mice, flies, as well as in mice and flies, we can measure, and we do see improvement in sleep quality. In humans, we do see self-reported improvement in sleep quality. So that might be the cause why.
Okay, thank you very much.
Sure.
We have another question from YouTube. I'm not really sure what it's related. It says, how long in hours is it TRF?
Yeah, so this is a question that comes up. I would say that time-restricted feeding is roughly, we can take the example of dental care, okay? So trying to eat everything that you eat with a healthy diet within 12 hours every single day is like brushing your teeth once a day, okay? So if you want to improve your dental health, you brush your teeth twice. So that will be TRF, 10 hours time-restricted feeding. And then you also do flossing once in a while, so that would be 8 hours time-restricted feeding. So that's how I look at it. If you're trying to lose weight or improve your, say, lipid panels, or if you try to prevent your pre-diabetes progressing to diabetes, then maybe 8 to 10 hours time-restricted feeding is important. If you're already healthy, you're athletic, you need a lot of energy input, then maybe 10 to 12 hours time-restricted feeding will be ideal. And if you're expecting, then you should not go below 12 hours of eating because of many obvious reasons. And of course, there is no study so far on pregnant women. So I can't say that time-restricted feeding will benefit or hamper women. But one thing that we see sometimes in our app is many people, particularly young women, they want to do everything at once, right? So they will do time-restricted feeding of 6 hours or 8 hours. Then they'll reduce their caloric intake. They will eat only one salad a day or a little bit of food, and then 5 miles. So that essentially puts them in negative energy deficiency, negative energy balance. And then they complain that they are losing their menstrual cycle, or they become irregular. So those are some of the signs that you may be going too extreme when you don't see normal endocrine function. So it may be convoluted answer, but then there is no, the bottom line is there is no hard and fast rule, but at least anyone from 10-year-old to 100-year-old must try to eat everything within 12 hours window. Which is not easy, actually, if you try it.
Thank you for your talk. Have you investigated whether time-restricted feeding has a long-term epigenetic modifications?
Yeah, we haven't tested that, whether it affects DNA methylation or other things. The one thing that we did was to see whether there is a legacy effect. So mice that ate time-restricted feeding, if we release them to ad libitum, are the benefits sustained, at least at the physiology level, whether it's blood cholesterol or blood sugar or even body weight, there is no legacy effect. They continue to gain weight because, so then the question is, even though you'll find a epigenetic change, what does it actually mean physiologically? It might mean a good publication for me, but in terms of connecting to that physiology and what people would like to see, I don't see a connection yet. Having said that, we haven't gone to deeper understanding. So for example, does time-restricted feeding changes something in the immune system that has a long-lasting effect? And in that case, in those cases, the epigenetic alterations will be important. So the bottom line is we haven't comprehensively looked at it, but it's a good thing to look at.
don't you think that the liver or the hypothalamus will be a good target for this modification?
I won't go for the liver because, the liver cells recycle in every 3 to 4 months.
Yeah.
So it would be...
But this recent paper that they just make therapy with epigenetic modifiers, they change the genome and they last even after regeneration.
Yeah. So we'll see how, like if the stem cells are affected and there is epigenetic changes in the liver stem cells or progenitor cells, yes, that'll be good to check. Yeah.
Thank you for your talk. Can you hear me? Okay. You showed that white adipose tissues showed the greatest amount of differential expressed genes.
Yeah.
Do you have any hypothesis why these specific tissues are more responsive to the intervention?
Yeah, so actually, this is done bulk RNA-seq. So one question, underlying question is, are we seeing changes in tissue composition, cell composition? So for example, are those big changes driven by immune cell infiltration state or immune cell polarization, which may have little to do with adipocytes themselves? And we do see that time-restricted feeding reduces macrophage infiltration in the adipose tissue in H&E staining and functional staining, so that might be one. And then the other one, if you think about it, adipose tissues do related, where adipose tissues, the fat metabolism can produce both pro-resolving and anti-resolving molecules, which are essentially pro-inflammatory or anti-inflammatory molecules, which can act as chemoattractant, chemorepellents, and also immune cell function modulators. Now the other thing is what we don't, we also didn't talk too much about is adipose tissue is heavily innervated by sympathetic nervous system, and there is adrenergic signal. And we know that adrenergic tone changes under time-restricted feeding. So whether there is also, neuronal adipocyte interaction that is driving some of the changes. But these are, hypothesis-generating questions or results. So thank you for asking that question too.
Yeah, thank you very much.
Hi, it's me again. Maybe it is really simple, but there's a thing that I don't really understand. Those circadian cycles age, I mean, even if you have the best lifestyle ever, your whole life, when you get old and experiment all these hormonal changes and that stuff, how they can affect the circadian cycle, even if you continue to have this great lifestyle. So how they can dysregulate.
Yeah, so whether in old age there is intrinsic change in circadian rhythm, right? So that's what you're asking. That's a very active area of research. People are trying to figure out where does that affect. So conversely, you can ask the other question that we know there are 9, 10, 11, 12 hallmarks of aging. Those things change with age, irrespective of whether it's a human, mouse, C. elegans, or even in yeast. So then the question is gradual accumulation of mutations in a cell, mutational load affect circadian rhythm, or is there any connection between telomere shortening and circadian rhythm, or proteostasis and circadian rhythm disruption? So we can ask that way. And again, as I said, this area is very open. What we know based on human cadaver tissue sample data that was published last year, I think, by Felix Neff's lab, is yes, in older individuals, there is dampening of circadian rhythm in many tissues. But at the same time, there is some reappearance of circadian rhythm. So that gives us some idea that there is not only in mice, not only in laboratory conditions, in real life, in older individuals, there are these changes. Now the question is, what can we do with lifestyle? To what extent we can preserve circadian rhythm? And I would say those experiments have not been done because it's, even in the US, it's expensive to get older mice and to monitor them. We have gone up to maybe 24 months, but not beyond that. Coming back to the other thing, can time-restricted feeding or many of these interventions will reduce the burden of cancer and many other diseases that are usually the cause of deaths for mice? The answer is no. They are not actually reducing that tumor burden. So bottom line is we don't know to what extent we can reduce the risk of disease in older individuals.
Thank you so much.
Thank you.